Hello everyone, welcome back to the NEET Master Series. I am thrilled to have you join us for our second video. And today we are focusing exclusively on MCQs from the chapter 5, that is molecular basis of inheritance, which is crucial topic for our NEET exam. Okay, so let us begin with our first question. Okay, what does lac refer to in what we call the lac operon? Option one is the number 1 lakh, lactose, lactase, lac in insect. So answer will be lactose. Lactose operon in E. coli is a catabolic pathway in which the structural genes remain switched off unless the inducer, that is lactose, is present in the medium. Second is DNA fingerprinting refers to molecular analysis of profiles of DNA samples, analysis of DNA samples using imprinting device, technique used for molecular analysis of different specimens of DNA, techniques used for identification of fingerprints of individuals. So answer will be A, molecular analysis of profiles of DNA samples. And DNA fingerprinting is the technique of determining nucleotide sequences of certain area of DNA which are unique to each individual. Okay. Our third question is genes that are involved in turning on or off the transcription of a set of structured genes are called operator genes, reductant genes, regulator genes, polymorphic genes. Answer will be A, operator genes. Operator gene allow the functioning of the operon. Our fourth question is transcriptional regulation in prokaryotes can occur by a repressor binding an operator and preventing transcription, an activator binding upstream from a promoter and positively affecting transcription, different promoter sequences binding RNA polymerase more tightly, resulting in more effective transcription initiation, or, or D option is all of the above. So answer will be 4, all of the above. Our fifth question is, in most eukaryotic cell, most genes are not expressed. This suggests that most eukaryotic regulatory protein act as exons, repressors, introns, activators. Answer will be D, activators. Sixth question is, lactose operon produce enzymes beta-galactosidase permease glycogen synthetase, beta-galactosidase permease transacetylase, permease glycogen synthetase and transacetylase, beta-galactosidase permease and phosphoglucose isomerase. Our answer will be B, that is beta-galactosidase permease and transacetylase. Seventh question is the most common way of gene expression is regulated in both prokaryote and eukaryote is through the control of mRNA translation, breakdown of protein formed by translation, prevention of DNA uncoiling prior to transcription, control of gene transcription. So answer will be D, control all gene transcription. So explanation is all of these are ways that gene expression can be regulated, but transcription control is clearly the most common mechanism. Okay. Eighth question is in prokaryotes, gene regulation occur at the level of transcription, translation, post-transcription, post-translation. Answer will be A, transcription. Okay. Ninth question is which of the following findings derived from recent analysis of the human genome does not illustrate our genetic relationship to other more primitive organisms? Our answer will be D, that is there are over 40 newly identified disease genes. 10th question is determination of one amino acid by more than one codon is due to redundancy of genetic code, continuous nature of genetic code, punctuation in genetic code, universal nature of genetic code. Answer will be A, redundancy of genetic code. 11th question is transcription in prokaryotic cell is initiated at a promoter using one of three RNA polymerase, that is RNA polymerase 2, initiated at a start codon with the help of initiation factor and the small subunit of the ribosome, initiated at a promoter and uses only one strand of DNA, The template is strand to synthesize a complementary RNA strand, and D is terminated at a stop codon. Our answer will be C, that is option A, describe transcription in eukaryotic cells, and option B describes translation. Okay, A describes transcription and B describes translation. So our correct answer will be C, that is initiated at a promoter and uses only one strand of DNA, the template strand, to synthesize a complementary RNA strand. Okay. Translation of messenger RNA into protein. Answer will be B, in 5' to 3' direction and from N terminus to C terminus. C, in a 3' to 5' direction and from C terminus to N terminus. Yes. Our 13th question is, a small segment of DNA contains the base sequence CGT. If an mRNA transcript is made that includes this DNA sequence, what will be the anticodon on the tRNA that will bind to the corresponding mRNA codon for this DNA triplet? Option A, CGT, GCA, CGU, GCT. Answer will be C, that is CGU. A DNA triplet of CGT would yield an mRNA codon of GCA. Okay, the complementary tRNA codon anticodon would therefore be CGU. A functional piece of mRNA has 66 codons. What is the maximum number of amino acid that could be present in the protein coded by this mRNA? Answer will be C, that is 65. Because a functional strand of mRNA must have a start and a stop codon. The start codon often also codes for the amino acid methionine, right? That is AUG codes for methionine, which is a start codon, right? This AUG is a start codon, which may or may not end up being a part of the final protein. However, the stop codon would not code for an amino acid. Thus, with 65 codons in the mRNA, there could be as many as 65 amino acid in the protein product. Our 15th question is, the primary function of DNA polymerase is to add nucleotide to the growing daughter strand, seal nicks along the sugar phosphate backbone of the daughter strand, unwind the parent DNA double helix, prevent reassociation of the denatured parent DNA strand. Answer will be A, add nucleotide to the growing daughter strand. 16th question is, the lagging daughter strand of DNA synthesized in what appears to be the wrong direction. This synthesis is accomplished by, answer will be D, that is A and B, because ligating connecting short Okazaki fragment that are synthesized in short splits in the right direction, and B is primase. Okazaki fragments are short pieces of synthesized DNA. The production of each of these fragments is dependent on the beginning RNA primer. The small fragments are ultimately ligated, that is connected together to form the lagging strand. Okay. 17th question. RNA primers are necessary in DNA synthesis because, answer will be A, DNA polymerase can only add to an existing strand of nucleotide. DNA polymerase cannot initiate the building of a nucleotide strand. It can only add to an existing strand. Thus, RNA primers are necessary to begin DNA synthesis. Our 18th question is, DNA replication is in process and energy. Hexagonic doesn't require, endothermic does require, endergonic does require, endothermic does not require. Answer will be C, that is endergonic does require. Its explanation is DNA replication is an energy consuming process that must have an input of energy to proceed. Energy is provided in the breaking of the triphosphate tails of each nucleotide. 19th question, which of the following statement about DNA replication is false? Answer will be A, that is, Okazaki fragments are the initiators of continuous DNA synthesis along the leading strand. Our last question is the key finding of the Hershey and Chase experiment on the mechanism of viral replication was that protein, not DNA, is the hereditary material. DNA, not protein, is the hereditary material. Protein and DNA play an equal role in determining inheritance. Neither protein nor DNA play a role in determining inheritance. So answer will be B, that is DNA, not protein, is the hereditary material. Hershey and Chase used radioactive labeling of sulfur and phosphorus to clearly demonstrate that DNA and not protein is the material that carries hereditary information. Okay, DNA is the one which contains hereditary information. So this is our second video, and this will be continued in further parts. So thank you guys, and make sure to like, subscribe, and turn on the notification bell so you never miss any of our NEET practice series. Let's dive into these MCQs and ace the chapter together. Thank you so much for watching.